Okay, hi everybody. We're going to start one of the last lectures before the final. So today we're going to talk about the support systems. So there are two fluid systems that support the neurons and the glial cells in the nervous system. So the first one is a cerebrospinal fluid system, which includes ventricles, the meninges, and the cerebrospinal fluid itself. And then we also have the vascular system, the blood supply. So the arterial supply, the veins and venous sinuses, and the mechanisms that regulate the flow. So first, I know we spoke about cerebrospinal fluid earlier in the course, but just as a review, the function of the cerebrospinal fluid is it supplies water, amino acids, specific ions, vitamins, and proteins to the extracellular fluid. And the cerebrospinal fluid also removes metabolites from the brain. This fluid also cushions the delicate neural structures of the nervous system. It offers the brain buoyancy and protection of the nervous system. It helps to absorb shock when the head is struck. So the picture on the right, you can see the ventricles in blue. Those are critical areas where the cerebrospinal fluid is both created as well as transported through. So you can see the big lateral ventricles. You can see that interventricular foramen that leads down to that third ventricle, the cerebral aqueduct, and then the larger fourth ventricle at the bottom. And then you'll notice below that is that central canal, which we know runs through the spinal cord. So that's kind of where the cerebrospinal fluid gets down into the spinal cord. And again, the cerebrospinal fluid is clear. It's that colorless liquid. It bathes the external surfaces of the brain and the cord. When we talk about the buoyancy of the cerebrospinal fluid, it providing the brain with buoyancy, it kind of unweights the brain. So the brain isn't like lying on the base of the skull. So, okay. So the flow of the cerebrospinal fluid. So it flows from those two lateral ventricles, one in each of the cerebral hemispheres. It flows down through that interventricular foramina into the third ventricle, then down through that cerebral aqueduct into the fourth ventricle. And that fourth ventricle is continuous with the central canal of the spinal cord. So there are openings in the roof of that fourth ventricle that lead to the subarachnoid space of the brain, which is also continuous with the subarachnoid space of the spinal cord. So that's the basic flow, and then it continues around. So let's continue to look. Okay, so we also need to talk about, when we talk about the subarachnoid space, so you remember early on in the class we talked about the meninges. So the meninges are three layers that cover the brain and the spinal cord. And those three layers are the dura mater, the arachnoid layer, and then that pia mater. You see the picture on the right, kind of is a close-up picture of those different meninges. So the dura mater, again, has an outer layer and an inner layer. So the dura mater has kind of two layers. That outer layer is bound to the inside of the skull, and the inner layer of the dura mater attaches to the arachnoid layer. And most of the time, these two layers are fused, and they are essentially one layer. But at certain places called the dural sinus, those two layers are not fused. And the sinuses are spaces for collection of the venous blood from the brain, that deoxygenated blood coming out of the brain, as well as cerebrospinal fluid. So that's where kind of deoxygenated blood and used, kind of, so to speak, cerebrospinal fluid is collected. And it's blue in that picture to the right. And again, that inner layer of the dura which is the one that's attached to the arachnoid layer, you can see that, has these, so the inner layer of the dura has projections. One of the projections is the falx cerebri, which separates the cerebral hemispheres. And the other big projection, remember, is the tentorium cerebelli, which separates the cerebellum from the cerebral hemispheres. So that dura is also kind of in the middle of the brain, separating sections of it. And so the arachnoid is a more delicate membrane, more delicate than that firmer dura mater, and it's attached loosely to the dura mater. And the projections from the arachnoid layer, you can see that arachnoid granulation. So that projection from the arachnoid layer protrudes and pierces up through the dura, and it allows the older or the used, so to speak, cerebrospinal fluid to flow back into the sinuses. So that's where the cerebrospinal fluid gets back into the sinuses, so it can be collected with the venous blood. We're going to talk more about that in a second. So you've seen these slides before. These are the slides from the meninges, similar to the ones we saw early on in the class. On that bottom right picture, you can see the falx cerebri and the falx cerebelli, and those are again the projections from the dura mater that separates different sections of the brain from each other. And again, the three layers of the meninges cover the brain and are continuous to the spinal cord, and they're part of that protective, part of the protection system for the brain. So we've talked about most of that. Okay, so you've already seen that, but I put these slides back in to kind of tie back to the earlier part of the course. Again, you've seen this picture again of here on the right. This picture is again another picture of the arachnoid villi. So these little pieces of arachnoid, these little projections kind of form together into arachnoid granulations, and that's how the cerebrospinal fluid gets reabsorbed into the venous blood, so it can come back out and to be kind of recycled, which we'll talk about in a minute. So, okay. And again, the pia mater is that innermost meningeal layer that kind of sits on the brain tissue. It's the one that kind of looks like saran wrap. It follows the contours of the brain, the grooves of the brain. And we'll talk about the meningitis in a minute. And again, you've seen this gross picture of kind of a gross anatomy picture of the brain, of the dura mater, just so you again can see it in situ, seeing it live. So the formation of the cerebrospinal fluid, the cerebrospinal fluid itself is made in the ventricles, in all the ventricles of the brain. So on the picture on the right, you can see these choroid plexuses, those little red arteries that picture is pointing to in the center of that ventricle. So the cerebrospinal fluid is made by these specialized capillaries from the pia mater. They're called choroid plexuses that project into each of the ventricles. In the two lateral ventricles, the third and the fourth ventricle. So you'll see a couple different areas of the little red choroid plexus. It's formed from the blood. So it's formed from the body's blood through filtration, through active transport, and facilitated transport through three layers of cells. So the CSF is formed through filtration as it goes through the capillary wall, the connective tissue, and the endothelium. And again, cerebrospinal fluid is similar. It's a fluid, and it's similar to kind of blood plasma. So except at the choroid plexus, the ependymal lining is freely permeable. So we're going to talk about why at the choroid plexus, at the location where these small capillaries are, why they're not freely permeable. And the ependymal cells are those, remember we talked about those as a neuroglial cell. The ependymal cells line the inner lining of the ventricles, and they're snugly spaced, and they're joined by these tight junctions over the choroid plexus.